Okay, good afternoon, everybody. I'm going to try this afternoon to finish the section that we're doing on DNA and DNA use in forensic science. As I've said before, probably one of the most important topics for you to know something about, because it has really revolutionized forensic science. So just to remind you where we finished off last time, we were talking about the polymerase chain reaction. And in polymerase chain reaction, we actually make use of the natural properties of the DNA molecule, which enable it to be very easily replicated. And remembering that the DNA molecule is a double molecule, the two sides match one another, and they match A to T and G to C. Those are nucleotides that match one another. And we can separate those two strands and bring in nucleotides which match the old strand. So we end up with an old strand with a new strand attached to it on both sides. And we've ended up doubling up the DNA molecule. So in the polymerase chain reaction, PCR, which is performed in a PCR machine these days, we put in a sample of DNA and we heat it. And as we heat it, it melts. The bonds between the two sides of the molecule break and the two sides separate. We then allow a primer to bind to each of the strands, and I'll return to that in a second. We first will attach a primer to each of the strands. Then we provide lots of nucleotides and also a DNA polymerase. And we allow the DNA polymerase to run along the molecule and duplicate each of the strands. And we end up with two molecules where we had one before. And we repeat that cycle over and over again, heating, cooling, heating, cooling, heating, cooling, etc. And when we have run this like between 30 and 35 times, we end up with an enormously amplified amount of DNA. But it's not all of the DNA. It is a specific area of the DNA that we have chosen. And we choose that piece of DNA that we amplify by designing our primers, by carefully designing our primers to sit at a particular place at the side of the area that we want to amplify. So this is the clever trick in polymerase chain reaction. We actually isolate an area of the DNA and we amplify that. We just replicate that area alone over and over and over again until we have a high concentration of that section of DNA that we can then analyze. We can then have a look at. Very quick. It's actually become a very, very simple technique. And if you carry on in biology, studying biology at some point or another, I'm sure you will have a chance to run PCR of your own. So, hold on, it's me. So here's its particular application that is so important to us. We talked before about RFLIP, restriction fragment length polymorphisms, the first kind of DNA fingerprinting. RFLIP depends, it's a very good technique, it's very accurate, and it provides a high statistical probability of individual characterization. The problem with it is you need a lot of DNA because the actual restriction fragment length is very, very long. It's also looking at repeating sections of DNA, but they themselves are quite long. And they repeat thousands of times to produce a very long area of DNA. Now we can analyze that by digesting it out and running them on gels, etc., etc. It's a long, tedious process, but it is extremely accurate. Its huge disadvantage is RFLIP, restriction fragment length polymorphism analysis, needs a lot of DNA, and it is difficult to run from a small sample collected at a crime scene, or also from a very old sample, because the great length of the restriction fragment length, it breaks rather easily as as the DNA ages. Instead, nowadays we make use of another kind of characteristic of DNA. It also depends upon tandem repeats. That is, sections of DNA where the DNA sequence repeats over and over and over and over and over again in segments. But this time, first of all, this repeating segment is very short, often 4 or 5 nucleotides or nucleotide bases long. And they don't repeat that much. So the whole length of this area of DNA is up to about 500 nucleotides long. So it's short. There are the second advantage. There are many, many, many of these areas in our whole genome, scattered throughout all of our chromosomes. There are areas where there are segments of repeating DNA sequence. We refer to these as short tandem repeats to distinguish them from restriction fragment lengths, because they're only up to about 500 bases in total. And if we go to these different sites all over the genome, we find these things scattered throughout the genome, and they have their own characteristics. They have different nucleotides, short nucleotide sequences. One. Second of all, they have the short tandem repeat segment is bounded on either side by specific DNA sequences that we know about. These have been well studied by geneticists. So we know what the DNA sequence is on either side of each of these different short tandem repeat areas. So we can design primers to amplify specific short tandem repeat areas. We can go to various places in the whole genome. Using our specific primers that we build, we can start the replication process at those specific areas and only amplify the short tandem repeat area that we are interested in. So the first thing is, these are very numerous throughout the whole genome. We don't know what their function is. They're not part of the genes. They just sit in between the genes. We don't really know what their function is. Most of them, the repeating segments, only 3 to 7 bases, and often it's 4 or 5. And together, they don't repeat that often, and they have spaces in between them. And altogether, they're just about up to 500 bases long, many of them far shorter. So they're little tiny things. That makes them very suitable for PCR. PCR works very well on these short lengths of DNA. PCR would not work on a restriction fragment length polymorphism. The restriction fragment length is too long to run it through PCR. You could run part of it, but you can't run the whole thing. But short tandem repeats are short enough that they're very amenable to polymerase chain reaction. Okay, so that's number one. They're very numerous. Each of them has their own characteristics, okay? The repeating nucleotides at one site are different to the repeating nucleotides at an entirely different site. So you've got a lot of variability there. Second of all, or maybe third, I can't remember at this stage, the number of times they repeat is very variable. In the whole human population, you go and you look and you see that different people have the same repeating segment a different number of times. That's point number 3. Point number 4, you inherit one DNA molecule with its short tandem repeat segment from your mother, one from your father. They can be the same, but they can also be different, and they very frequently are different. So when we go and and amplify them with PCR, we very often find we have 2 fragments produced, one from the mother, one from the father, with the same repeating segments, the same repeating bases, but repeated a different number of times. And so you can have the character of that site from your father may be different to your mother. So this means that there is a certain amount of variability, which is very well characterized for each of these single tandem repeat sites or loci. There, it's called a place on the DNA where something is located is called the locus. So for any specific short tandem repeat, it has a locus in the genome, a place where it occurs. So we know a lot about them. They have sampled many people throughout different populations. There are some ethnic differences, but by and large, we know what the chances of inheriting one particular locus is. And we can work out what the chances are if it's just of inheriting a particular maternal and paternal pattern together. We can work that out statistically as well. So these are very useful, extremely useful. Don't forget that we don't only look at one. Right? For any one site, the statistical chance of inheriting it is low compared to having it just happen by chance. But we've got many of them. We've got many different short tandem repeat sites throughout the genome. And if we look at a lot of them, we begin to add together the probability. So for maybe we just look at one site, okay? And we say, well, what is your chance of inheriting that purely by chance? And we work it out for the whole population. And we say, the chance of inheriting that is 3.5%. Okay, that's 3.5% chance that you inherit that site just from the general population. Well, that gives us a ballpark, but it's only a ballpark. It's not individual character, it's class character. But there are many of these STR sites. So when we begin to look at the chance of inheriting this one, together with another one, together with another one, we have to start multiplying the probabilities together. And the probability of inheriting a particular pattern, which is identical with all of those sites, becomes very, very low, very, very quickly, as we'll see in a minute. So let's have try and picture in our minds what these actually look like. So here we're going to a site. Here's the repeating segment there. Let's just say it's, I don't know, 4 or 5 nucleotides long. Here it repeats again. Here it repeats again. This here, this whole thing here is one DNA molecule, okay? So here we have our nucleotides repeating. Here are their matching repeats, repeat. 3 of them, 1, 2, 3. Flanking this site here, this is the locus there. That's one particular locus, and it will be given a name. If it's been characterized, it will be given a name, as we'll see in a minute. Flanking it, there is DNA sequence, which we know. Here it is, CCAG on this strand, and here it is on this strand here, GAAT. Okay, we design a primer. The primer is going to sit there when we start. Here's our other primer. It's going to sit here. What are we going to do now? We're going to put this through PCR, through polymerase chain reaction. We're going to melt it, going to heat it up. It's going to separate. We're going to replicate that, and we're going to replicate that. Okay? Now, cool it, and now we're going to heat it up again. We're going to do it again. What are we going to do? We're going to end up amplifying to a high concentration this segment here. Just those 3 repeating segments. This whole thing here is going to be built up to a high concentration of DNA. It's going to select that out of the whole genome. The rest of the genome is not going to be replicated. We're just going to replicate this and and get it to a high concentration. Remember what I told you. This is one DNA molecule. Maybe you inherited this DNA molecule from your mother. This is one chromosome. Remember, chromosome is one DNA molecule inherited from one of the parents. So maybe this is from your mother. You've got another one from your father. Well, the other one from your father, it'll have the same, it's the same nucleotide sequence in the repeats because it's the same locus. But it may have a different number of repeats. Instead of 3, it may have 5 or 8 or whatever. We know for each locus, we know what the characteristics are that have been discovered in the human population. So we can work out what the probability is of you inheriting just this from your mother, or we can work out what the probability is of you inheriting a particular pattern from mother and father. There is a statistical likelihood that you would inherit that. And it also gives us a statistical likelihood that a DNA sample that had the same structure could be arrived at by chance from the general population. So we say you have a 3.5% chance. That means if we went out and we sampled 100 people and looked for this particular site, 3.5% of the population would have that particular pattern, right? So that means that just for one site, it would only give you class evidence. It would be able to exclude people who didn't have this pattern, but you would be included together with the people, with other people. It just tells you that that's a class. But remember what we said, there are many of these sites. And as we work out the statistical probability of inheriting 1, and then 2, we multiply their probabilities. 3, we multiply the probability. The more we have, the smaller and smaller and smaller the chance becomes that you could inherit that by chance from the general population. That a sample of DNA taken from a crime scene, for example, matched to a suspect purely by chance is vanishingly small when we start to look at many of these sites together. So here's just one site, okay, and 3 here, and we don't yet know what the paternal one would be. So just in general, these single tandem repeats between 3 to 7 bases, often 4 to 5 in length, each little repeating segment. And the whole thing usually less than 500 bases in length. That gives us the advantage, first of all, as I've explained, that it's very easy to subject them to PCR. But it also means that they survive very well. And these techniques can be done, these PCR techniques can be done on very small DNA samples, what we refer to as touch DNA, for example, just 1 or 2 cells left behind in a fingerprint, for example, which is very easy. It would happen the whole time. In addition, we can derive the sequences from DNA very often, which is old, it has dried out, it's been exposed to sunlight and to the air, et cetera, for some time. So let's have a look at a specific one. And this is one which is often used. It's called TH01, and some people call it THO1, just because that's what it looks like. It's actually TH01. And its repeating sequence is AATG. So there are 7 variants of this, which have been seen in the human population that has been studied. It's not to say that others don't exist. It's just that 7 of them have been discovered. And the variability is, the variants contain between 5 and 11 repeats of this AATG. So on any one molecule, looking at the whole human population, you could have between 5 and 11. That's where our 7 variants come from. 5, 6, 7, 8, 9, 10, 11. That's 7 variants. So we're going to have a look now at a specific case, okay, a specific individual. And this individual has two variants, one inherited from the father, one inherited from the mother. And the one variant has 6. The other variant has 8. So let's just say 6 from the mother on the mother's side, 8 from the father. What we're going to do, let's say we have a crime scene and we have a questioned stain, a semen stain, let's say. And then we have a suspect. So we're going to go and we're going to run the PCR on the questioned stain, and we're going to amplify. So the THO1 locus, the THO1 site is going to be amplified. Well, when we do that, we're going to amplify the maternal and we're going to amplify the paternal at the same time. In this case, there's 6 on the mother, the maternal side, 8 on the father, on the father's chromosome. So we're going to end up with 2 bands. It is possible that the paternal and the maternal could have been the same, in which case we would just have 1 band. But very often they are different. And in this case, they're different. Maternal 6, paternal is 8. So we're going to amplify our THO1, and then we're going to run it out on gel electrophoresis. As I described before, we're going to run it through a gel, which is going to sort the fragments by size. And this is what our 2 molecules look like. Here, 1, 2, 3, 4, 5, 6. Let's say this is maternal. And here, 1, 2, 3, 4, 5, 6, 7, 8. This one, obviously, is much shorter than this one. We're going to amplify this with PCR, and we're going to end up with a high concentration of DNA. And when we run it out on the gel, here's what we find. So first of all, this is a real crime scene. This is for the THO1 site. And first of all, here's this side here. These are molecular markers here, just to tell us what size we're looking at. And 1, 2, 3, 4, 5. So here is the questioned DNA here. That's from the crime scene. And we've amplified just the THO1 site. And here from that stain, we obtain 2 bands. And this one here is shorter. So this is the 1, 2, 3, 4, 5, 6. That's the 6th band. This one here is longer. It runs more slowly, so it's longer. And that is the 1, 2, 3, 4, 5, 6, 7, 8 band there. So here's suspect 1. Suspect 1 is interesting because they only have 1 band. And that is because both the mother and the father have 6. So both of them run together, and you can't tell one from the other. You just have 1 band. Here's suspect number 2, and look, suspect number 2 matches there. Okay, here's the 3rd suspect, and they're completely different. They have a longer than 8 and much shorter than 6. And so they don't match either. So here is actually our suspect matches. But take this to a court of law, and they're going to ask you, well, how many people in the population have THO1 and that pattern, 6 and 8? And the answer is about 3.5%. That means if we sampled 100 people, let's say we sampled 1,000 people, right? Just to get the numbers right. 1,000 people, 35 of them would have this pattern, 6, 8 at THO1. It's good class evidence. It allows us to say, look, first of all, we can exclude everybody who doesn't have that. But it only allows us to put our suspect into a group. But don't forget, we don't only have THO1, right? We have THO1, we have in 3.5% of the population. But there are many of these short tandem repeat sites, many of them, and we can sample more of them. And every time we sample another one, we take its probability and we multiply it times 3.5%. 3.5% is 0.035. That is the probability that you would inherit that just from the general population, that you would find that from the general population sample. The next one may be 0.35 as well. Well, if we do 2 sites and each of them has 0.35, to work out the probability of having those 2 by chance from the general population, multiply 0.35 by 0.35, becomes very small, and becomes very small very quickly. And well, this is exactly what we do. We actually go to many of these short tandem repeats sites and characterize them. And the more we do, the smaller and smaller and smaller and smaller the probability is that our question sample and its match occurred purely by chance from the general population. The smaller the probability that that match occurred by chance from sampling the general population means the higher and higher probability that our match is an individual match, that this question stain from the crime scene matches to the suspect because the suspect deposited the question stain. It may not give you much, but just looking at one, but when we look at 2 sites, 3 sites, etc., the probability it occurred by chance becomes very, very small. So we don't have to do this individually. In fact, we can run them out because of the wonder of using different primers in doing our PCR. We can put in several different primers into the same PCR run and amplify different areas of the genome as long as we know that they are very different in size. So what I mean by that, this is called multiplexing. So here are 3 different short tandem repeat sites which are used. These are familiar sites to a forensic geneticist. They're called FGA, VWA, and this D3S here. So first of all, you'll see they're very different size. This never gets bigger than this. Okay, this never gets bigger than this. So we can run them out at the same time and still separate them. And here we're running out the 3 sites. The chance of getting this by chance from the general population, who knows, 1 to 3% or whatever. The chance of getting this by chance, we don't know, 5% or whatever. But to inherit that pattern and this pattern at the same time, multiply the probability here by the probability there, it starts to get low. Do it for 3. You have to multiply the probability there by the probability by the probability. The probability of inheriting all 3 by chance from the general population is very, very low. It gets very low very quickly. And so this is what we actually do. We actually look at many different sites, and currently we actually look at 20. It used to be that they looked at 13 different SDR sites. This is when this technology first became available, and they started plugging it into this database called CODIS. And CODIS is the database for lodging DNA information for forensic purposes. In 2017, this was increased to 20 different sites, and you'll see the impact of that in a second. By using those 20 sites, we arrive at astronomical numbers, where it becomes 1 in trillions that a sample and a standard were matched by chance. It's just infinitely small that could have happened by chance. The huge advantage of using these short tandem repeats PCR technique is you only need a very, very small amount of DNA, extremely small. And this has given rise to the ability to develop what we call touch DNA. As I mentioned before, simply handling an object, for example, it deposits cells with DNA in them on the object, and DNA can then be extracted and amplified by PCR. So here are some of the actual statistics. This is the old statistics. This is for 13 sites. Realize that with 20 sites, it's enormously increased. So if we go to the SCR site, and if we were to match the three that are commonest in the population, we look at a sample which has a pattern for those three, and then we look at a standard from a suspect or whatever, and we would see that for those three, if they all matched, well, 1 in 5,000 people have that combination. 1 in 5,000. This is not individual yet. This is not really good enough. There are 5,000 people. 5,000 people is not that many when you're trying to pull out one suspect who committed a murder or a rape or something like that. It is possible. It is distinctly possible that you got a match by chance from this population of 5,000 people. However, when you match 6 different single tandem, I keep calling single, short tandem repeat segments. When you match 6 short tandem repeat segments, it's 1 in 2 million people carry that, those matching 6, beginning to get very remote. Match 9, and you're talking about 1 in a billion people. Match all 13. Now, at this point, I need to remind you, there are some ethnic differences. Different populations have different rates of occurrence, so you need to take that into account. But if you match all 13 sites between a question and a standard, the chances are 1 in 575 trillion Caucasian Americans would carry that combination. 1 in 900 trillion African American. It gets now to literally astronomical numbers, because this is like the number of stars that we have in our galaxy. So the probability when you get this kind of match between a question sample, like from a crime scene, and a suspect, this is truly damning evidence that the two were deposited by the same person, because you'd have to search through 575 trillion Caucasian Americans to find another person that carried that, matched all 13. There are currently only 7 billion people on the planet, and about 100 billion have ever lived at all. So more people, this is a tiny fraction of 900 trillion. Okay, 100 billion people that have ever lived. It's a tiny fraction of these numbers here. This is a very powerful forensic technique for determining identity, for determining a match between question sample and the standard. This used to be done laboriously by hand and in the PCR machine, but then run out on gels and everything. It's no longer the case. Nowadays, it's done in automated fashion, and it is run through machines, which in fact can do it very, very quickly in a matter of hours. And this is a typical pattern that such a machine would produce. And here is each of these here is a different short tandem repeat site somewhere in the genome. Here's SO1 there, and you can see here there's 2 different bands there. Okay, and for each of these, now this gives us an SCR profile, and now we can go to a standard. Let's say this is a question sample. We can now go to a standard and do exactly the same thing and see how we match, see what the match is. And we can also at the same time work out what is the probability that that match would occur by chance just from the general population. If it's 1 in 575 trillion, you can be sure you are dealing with a common source for your sample. That is the huge power of this, and that is why so much attention is paid to DNA evidence in these cases where it occurs. I would add a caveat though, and that is the collection and technique of amplifying the DNA needs to be done very carefully. Very, very careful control needs to be exerted over both collection of the samples and of their running, because error can be introduced into, and that we must always be aware of. Okay, so just let me flip back here because there's something I want to show you. Okay, these are the 15 sites, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So each of these little blocks is a different site. But have a look here at this one here. Can you see there? It says X, Y. The reason for that is that this is, in fact, the sex chromosomes. Okay, and these are very useful, especially the Y chromosome, because Y chromosomes are only carried by males, by biological males. And the Y chromosome is inherited from your father, if you're a male. If you're a female, you don't inherit a Y chromosome from your father, you inherit another X chromosome. But the Y chromosome is very short, it's very small, but it does have short tandem repeat sites on it. In fact, it has quite a lot of different short tandem repeat sites on it. And a number of these have been characterized. And the reason that this is important is because it can become very, very difficult to sort out DNA samples which are mixed. So for example, if you had a violent incident involving 2 males, or even more males, you could have blood or whatever deposited by several of them, and run that out in PCR, and you're going to amplify everybody's DNA, and it becomes very difficult to sort out who is who in that situation. But these the Y chromosome STRs allow us to actually start to separate the samples and individuate the different males who might have been involved, because the likelihood is very, very small that their short tandem repeat sites on the Y chromosome would be the same. Again, we can work out the statistical probability, but it becomes a very useful technique for sorting out males who might be involved in the same crime. The other kind of DNA, which is starting to be made more use of, is a very interesting aspect to our genome. I've told you before that most of our DNA is lodged inside the nucleus of our cells. The vast majority of our DNA is in the nucleus of our cells, but we have these little organelles in our cells called the mitochondria, which are responsible for producing all the biologically useful energy that we use the whole time. They are the powerhouses of the cell. And very interestingly, they have their own DNA. It's in the form of a little circle of DNA. And they are able, is replicated by the mitochondria and independently of the nucleus. And it's another little store of DNA information that we can, in fact, make use of, but it's very short. That mitochondrial DNA genome is very, very small. What is interesting about it is that we inherit our mitochondria from our mothers, not from our fathers. And the mitochondrial DNA sequence, we share that sequence with all of our siblings that were born by our common biological mother. We share it with our biological mother. We share our mitochondrial sequence with our grandmothers, our maternal grandmothers, maternal great-grandmothers, et cetera, et cetera, et cetera. So this can be very useful, especially in determining identity, because we inherit the mitochondrial sequence through the maternal line. And if we can get, for example, if we're trying to identify somebody by using mitochondrial DNA, we can get a sample from a sibling and see if it matches. The other advantage of mitochondrial DNA is even in very, very old DNA samples, in very, very old samples of biological fluids, even in bone and things like this, mitochondrial DNA can be extracted and can be sequenced. And it can even be, sometimes has been recovered from burn samples and things like that, where all the other DNA has been destroyed. Mitochondrial DNA actually does survive. The other place where it is very useful is if all that we have is hair, because mitochondrial DNA is present in hair where nuclear DNA is not. There are some areas of the mitochondrial DNA which are very variable, but where we hope that we can eventually start to use those to provide individual characteristics. But in general, it only allows us to identify to the maternal line of inheritance. Okay, so CODIS is the database in which all of this information ends up being lodged from crime scenes, from samples taken legally in different states. The law is different. But in general now, wherever possible, authorities will take a DNA sample because it provides such a powerful individual identity. Those are all lodged in the Combined DNA Indexing System, or CODIS. And it's an FBI administered system. And it coordinates all the information from local, state, national databases of DNA offenders. As I've said, who ends up donating DNA to this DNA information to the database varies by precinct and it varies by state, etc. The law is still relatively unclear regarding DNA collection. Very often, for example, in the start, they made use of samples collected under medical procedures and things like this. It's no longer done. You have to give permission under those circumstances for your DNA to be used. If you have committed a crime and been arrested in some states, you can have your DNA taken. In other states, you have to have been convicted in order for your DNA to be lodged permanently in CODIS. And just remember that currently they utilize 20 short tandem repeat sites in the genome, which means if you match on all of those 20, it's 1 in hundreds of trillions. That is the probability that is unique. Okay, so where do we get DNA from? Well, nowadays from many, many different sources. It used to be blood, semen, and saliva were the only ones that provided enough DNA. But nowadays from skin cells, even a very, very few skin cells provide enough for DNA. It can also be extracted from hair if the hair is pulled out together with living tissue, the hair root, which very often happens in fights and this sort of thing. That will contain plenty enough DNA to be sequenced and typed. Otherwise, in hair, the rest of the hair does not contain much DNA except for mitochondrial DNA. In bone, especially in a long bone, for example, where cells may be protected from bacterial activity during the processes of decay. DNA may be extracted even from very, very old skeletons. The one place where it lasts pretty well, in fact, extremely well, is in the teeth. And very frequently, DNA can be isolated from the teeth, whereas it's completely decayed everywhere else in a skeleton. DNA has even been extracted from teeth of Neanderthals and Neanderthal fossils and this sort of thing. Remember, the DNA is a very strong molecule. That backbone that forms the molecule is very, very powerful. And that is what gives it its strength and its durability as well. So at a crime scene now, many, many things will be examined with an eye to the possibility of collecting DNA. Anything which might have been touched, glasses, cans, cigarette ends, chewing gum, etc., etc., etc. It can also often be extracted from swabs taken of bite marks, saliva mark, all of those sorts of things. Obviously, anything which a person has ever licked or anything like that is likely to contain enough cells for us to be able to amplify the DNA by PCR. Obviously, clothing, all of these sorts of things now are collected very carefully with an eye to the idea that they may have DNA evidence, both from a suspect and from a victim, for example. But if at a scene, if there are blood stains, paradoxically, what is usually done is that those blood stains are subsampled, usually with a Q-tip, with a cotton swab. Often, if the blood stain is dried, you can moisten the cotton swab, swab the blood stain, but the swab is then immediately dried. It's dried because drying is, especially rapid drying, is not going to hurt the DNA. It's not going to damage the DNA, especially if the DNA is going straight to a laboratory. But if you kept the DNA, if you kept that swab in a moist environment, very quickly bacteria would begin to degrade the biological material. And in doing so, they could degrade the DNA as well. That's the question stains. Question stains are usually dried, packaged very carefully, and transported as quickly as possible to the laboratory, if possible, under refrigerated, not freezing, but under refrigerated conditions. The standard swabs are taken under laboratory conditions, or under using a laboratory protocol, usually a mouth swab, which you would call a buccal swab. Quickly swirled around the mouth, it collects plenty enough loose cells that a DNA profile can be developed from that. As I say, these are the samples are usually dried to prevent any bacterial or fungal activity. But the location of these stains is very important, and the background information about their location and their extraction needs to be preserved as part of the chain of evidence. So they're very often the stains, etc. Before they are sampled, they are photographed, detailed in drawings, detailed in notes, etc., etc. Just give me 1 second. I'm sorry. So they are not put into airtight containers. They're usually packaged in manila envelopes. And if need be, they can be transported under refrigerated conditions. The critically important thing, and many investigators have made this mistake, is that it is very easy to contaminate a crime scene with your own DNA. It's another example of Locard's principle, right? Always bring, when we go to a crime scene, we leave stuff and we take stuff away with us. And what we leave may well have DNA associated with it. And there have been instances where evidence has had to be thrown out because it's been shown that it's contaminated with examiner's DNA. So all possible where DNA may be present and where it needs to be collected, all possible precautions are taken. To the extent now that people are seldom enter a crime scene ungloved. They may also wear eye protection. They may wear a surgical mask. They may wear a head covering, and may also wear booties, this sort of thing, all to avoid the possibility of contaminating biological samples with the investigators' own DNA. Their samples are never touched as far as possible. Instead, instruments will be used to pick up, deposit, move in samples, so that we avoid any possibility that contamination could occur. Okay, basic common sense precautions, obviously. All right, so with that, that's the last of our DNA section. I know that for some of you that some of this is a bit difficult to comprehend. So what I would advise you to do, go right back to the very beginning and understand, make sure you understand the structure of DNA. Its basic structure is a double molecule, how it is held together, the simple principles of how it is replicated, the importance of the primers, not only in biological systems, but the importance of the primers for us. Then try and understand this concept of different areas of the genome having segments of repeating nucleotide sequence. These repeating segments, very long ones, which are analyzed by cutting them out with restriction enzymes to give us restriction fragment length polymorphisms. And then these short tandem repeat segments, which we use 20 different loci in the genome, which we amplify by PCR. And the amplification of PCR, we design the primers that start the replication process so that we only amplify one specific area of the DNA that we are interested in. If you get all that put in as a story through your brain, you'll be able to do well on any questioning that a court presents to you or that I give to you in a test. Okay, so then I will see you on Wednesday.